Okay.
Cool.
Right. Welcome, everybody. Thank you all for coming. It's my pleasure to introduce Dr. Sam Berens. Sam did his PhD here in Sussex a few years ago, and he left us to go to the University of York to do a postdoc. And now he's just recently rejoined, so it's great to have him back, and it's great to now we get to hear about some of the work he did while he was away. So over to you, Sam.
Yes, thank you very much for having me. It's good to be back. I'm going to be talking about an imaging project that I ran as a postdoc in York with Aidan Horner, my then supervisor. And here we were really looking at how we're able to rapidly learn the layout of a new spatial environment. So obviously, when you go into a new environment for the first time, you can't possibly survey all your surroundings and sample the environment from every possible viewpoint simultaneously. So spatial learning in general requires us to integrate information across different views. And it's this kind of integration of different viewpoints that we looked at in this study. So just to start off by introducing a bit of background theory, I'll mainly be talking about this model of spatial memory and imagery from Burgess and colleagues, often referred to as the BB model or the BBB model because of the initials of the authors involved down here. And other models of spatial memory and navigation are available, I should say. However, I'm focused mainly on this because this captures shared features of those alternative models very nicely. And also it's by far the most well fleshed out with being a fully fledged computational model. And at the heart of this model is this supposition that the medial temporal lobes maintain an allocentric representation of one's location in space that is not dependent on the particular viewpoint that an agent is taking or their heading direction or anything like that. It's just encoding their spatial location in an allocentric frame of reference. So for instance, the parahippocampal cortex is thought to represent the boundaries of our current spatial environment, and the hippocampus is thought to build on those boundary-based representations to form a Cartesian representation of our current location in the space. And all sorts of different spatial cells are involved. So for instance, boundary vector cells encode our distances to the environmental boundaries. Place cells represent the lattice points in the Cartesian grid. There are also object vector cells, trace vector cells. There's this whole family of spatial cells involved. In contrast to that allocentric representation in the medial temporal lobes, there's also an egocentric representation in the parietal cortex. And this is viewpoint specific, so an egocentric representation. That's dependent on the heading direction. And obviously this is very important for route planning and kind of avoiding obstacles, because it's not just enough to have an allocentric kind of bird's eye view, if you like, plan of the environment. In order to plan a trajectory that avoids obstacles and navigates to a goal, you need to have, a representation of what you're seeing right in front of you. And that's what happens in the parietal cortex, according to the model. The retrosplenial cortex kind of mediates between these two reference frames. So in the retrosplenial cortex, it's thought that a set of approximate linear transformations can map egocentric information in the parietal cortex into allocentric representation in the medial temporal lobe. Those are the bottom-up kind of connections. And you also have top-down connections that translate allocentric information in the medial temporal lobes into egocentric representations in the parietal cortex. So you've kind of got this forward and backward flow of information. So what evidence is there for these different reference frames in humans? Well, previous studies have mainly looked at finding patterns of activity, patterns of bold activity that are specific to a particular location, but are not dependent on your viewing angle or viewpoint that you're taking in a given location. And in these studies in general, they find patterns of activity indicative of these allocentric representations in the retrosplenial cortex, most notably on the right, also to some extent in the parahippocampal cortex and in the occipital place area, the OPA. So for instance, in this study over here by Vass and Epstein, 2013, participants were shown images inside the scanner of various positions in a road grid network that was very familiar to them. And those images might have either been facing north, south, east, or west. And independent of whether they were facing north, south, east, or west, the authors identified patterns of activity that were stable and consistent for a specific position or location in that grid system, that road network, but didn't depend on the viewing direction. Similarly, the study also from the Epstein lab from 2015, participants were shown pictures of various different buildings that were taken from either the inside or the outside of those buildings. And they found stable patterns of activity that represented the building and the surrounding area itself, but didn't depend on whether you saw an inside or an outside image. And finally, this study by Robertson et al. 2016, they trained participants before scanning. They trained participants to recognize that, say, for instance, this door, this black door over here, was 180 degrees opposite in space to this blue car over here. And just got people to learn that kind of spatial relationship, and found that the patterns of activity encoded this panorama location itself, but didn't depend on whether you saw an image of the door or the car, sort of thing. And that those representations were seen in the parahippocampal cortex and the retrosplenial cortex. Sorry, no, that's not true. I can't see my screen. Hang on. In the retrosplenial cortex and the OPA, but not in the parahippocampal cortex. Okay, so this is the state of play as we got to it. But there are a couple of open questions. So firstly, we don't really know. There's some ambiguity as to what kinds of, whether these representations are truly allocentric or whether they're truly spatial in nature, I should say. Because in all of these cases, participants knew the locations fairly well. In the Robertson et al. Study, participants had been extensively trained on these panoramas before scanning. And in these other two studies, the participants were personally familiar with these locations. So they were university students and the images were taken on a university campus that they studied at. So for instance, someone would have known that these two images in the center here were taken at a cinema. I believe it was a cinema. And so, there might be some semantic information that is driving these patterns of bold activity here. We can't rule that out. Additionally, according to the BBB model, these patterns of activity, the allocentric representations should be evoked very rapidly upon entering an environment. So all you need to do is enter an environment, kind of very quickly take in your surroundings, and you should have a stable representation, an allocentric representation that will be maintained until you leave that environment. Yet, with this very extensive training, we don't know how quickly these representations emerge. So those were two questions that we had. A final question is whether these allocentric representations, these viewpoint-independent representations, depend on any kind of spatial task. So in all of these cases, participants were asked to do some sort of spatial memory task at the same time as seeing the images in the scanner. And so it could be that the representations that we're seeing in these regions actually depends on some sort of spatial processing that's a characteristic of the task and not of just passively viewing. Again, according to the BBB model, this forward and backward flow of information should happen automatically. It's just automatic updated through self-motion in an environment. And that's something that we still don't have any evidence from the human neuroimaging literature, at least. So those were the two open questions that we really wanted to attack. Do allocentric representations develop rapidly, or do they require extensive training? And here we define rapidly as like 60 seconds of exposure. So I'll show you the stimuli in a second, but they were shown videos that panned across the locations that they learned in 20 seconds, and they saw that 3 times, so 60 seconds of exposure for each environment. Also, are these allocentric representations evoked during passive viewing, or do they require some sort of explicit spatial task in order to be seen in humans? So those were our 2 main research questions of the study. Okay, so to test those, I say we, the PhD student in Aidan's lab at the time, Bardor, who's now a postdoc with Neil Burgess at UCL, went out to 2 cities in the UK that are relatively underrepresented in the student population at York. So we wanted to sample locations where participants were definitely not had definitely not been, and we're actually, part of our exclusion criteria is whether participants had been to any of the locations in our stimuli. So Bardo went to Sunderland and Middlesbrough, and when he got there, he just went into the town and took these 180-degree panoramas of various urban locations, showing traffic lights and buildings and car parks and stuff like this. And from each panorama, we did 2 things. So the first thing that we did was we took 2 endpoints from each panorama, one at the 0-degree position and one from the 180-degree position. Each endpoint spanned a field of view that was about 60 degrees. And we showed these endpoints to participants inside the scanner. We also showed these panoramas inside the scanner in a series of videos, 2 different types of videos. So in an overlap video, participants saw the 60-degree field of view starting at the no overlap endpoint. It started by showing that endpoint over there. The video then panned across and swept over to show the 90-degree position, of which would be the central region over here. At this point, the video cut and showed the 180-degree endpoint, and then panned across in the opposite direction to show the 90-degree position again. So kind of by recognizing that there's this visual overlap between those 2 video segments, participants could learn or infer that these 2 endpoints were from the same location. And hopefully that would actually increase the similarity, the representational similarity between them, if they're forming an allocentric representation of this location as a whole. In the no overlap condition, no overlap videos, something very similar happened. So we started off by showing the 0-degree endpoint, panned across to the 90-degree endpoint. So there's this starting at this railings, pans across to this Italian restaurant in the middle here. And then the cut is to now a completely different panorama. So the second video segment is from a completely unrelated location. And so there's no visual overlap in the center here. I can demonstrate each of these different types of videos. I have some videos to show. Please let me know if you can't see any of this. So first I'll show an overlap video. So pan across to the center. If you just remember, like, there's this pole and some baskets and stuff, and you can kind of see that come across here again. And the video sweep twice, I should mention that. So this is an overlap video. It's fairly brief, just 20 seconds. And then a no overlap video looks kind of a bit strange. Pan over. No visual overlap. It's a little bit trickier sometimes to see, but you can kind of see there's no overlap there. Okay, so those were the videos. Yeah. And so in all, we had 3 essentially three different conditions. Let me make this full screen again. So we had three different conditions. We had image endpoints. So these are the endpoints that were shown in an overlap video. And I refer to those as the image endpoint A1, which is the 0-degree position of an overlap video, and A2, which is the 180-degree endpoint of an overlap video. So A1, A2, this image and this image. We had endpoints that were shown in a no overlap video, B1 and C2. So they're from different panoramas. So that you've got B1 and C2 as the different letters refer to the panoramas. So this would be B1, this would be C2 over here. And you also had image endpoints that were not shown in any video at all. And they correspond to the endpoints that were missing from the panorama in this top row of the no overlap video. So the whatever is opposite this building over here, and also the corresponding one for the panorama below in the second video segment. So whatever is opposite this building here, and that would be C1. So we have these three experimental conditions: overlap, no overlap, and seam. And the hypothesis was that at first you would see no differences in similarity between these endpoints. They would all kind of be the same. And it would only be after participants had seen endpoints in an overlap video condition that the similarity between A1 and A2 would increase. The representational similarity as measured by RSA would increase, but you wouldn't see any corresponding increases in the other conditions. That was our hypothesis. So just to talk about the task structure, what happened inside the scanner, everything that happened inside the scanner is shown in these purple squares. So as soon as participants got in the scanner, they did a functional localizer to identify scene-selective regions of the cortex. This was a simple face versus scene localizer. The scenes were nothing to do with these Middlesbrough or Sunderland images that Bardell collected. We just got them from the internet, made them black and white, all the same size, all that sort of stuff. And images of faces were used as to contrast that. And then we showed the endpoints to participants in the scanner in a simple kind of slideshow presentation. The only task that participants had to do as they saw those images flash up one after another was to detect these oddball targets, which were 3 small red dots that were superimposed on top of a small subset of the images at random locations. So very occasionally, 1 out of 9, 11% of the time, participants would see 3 small red dots superimposed on top of the images. They had to keep an eye out for those targets. And every time they spotted those red dots, they just had to hit a button to indicate that they had seen it. We deliberately made this very difficult, or we deliberately made it difficult so that participants would have to scan the images quite intensively and made sure that they were paying attention to these images. And they did this red dot target detection task both before and after the videos. So over here and over here. The red dots were not this size, by the way. It's just to illustrate that, it was a red dot task. And in between those 2 red dot tasks, we also gave them these videos. So we just asked them we showed them the videos one after another. Each video was presented 3 times in a random order, and participants were asked to identify whether there was a visual overlap or not. So during the videos, there was a kind of a spatial task. Was there a visual overlap or not? Outside of the scanner, both before and after the scanning session, we gave participants a simple behavioral task. So in the behavioral task, one of these endpoints would have been shown in a red box as a Q. I have a better slide of this. One of the endpoints would have been shown in a red box as a Q, and the task was simply of a series of 5 alternatives that flashed up after that Q, to identify which of the alternatives belong to the same location as that Q. People just had to indicate with a simple button press. As I say, this happened before and after scanning. So we could gauge whether participants had kind of learned a spatial representation for each of those images, so had learned which images were from the same location. We also ran a similar task, which was really, really similar in terms of how it ran, but the question was different. It wasn't which image was from the same location, but which image was shown in the same video. So slightly different. Obviously, this only happened after, only happened after scanning. Yeah. So those are all of the tasks as they happened. So the behavioral data, I'll talk about the behavioral performance on those behavioral tasks first. Along the y-axis here, I'm plotting the probability of a correct match. So the probability that given a Q image, you can identify the target image, which was from the same location as that Q. Chance level, all things being equal, is at 0.2, 1 out of 5, because there were 5 alternatives. And you can see that before the videos were shown, everyone was at chance at doing this scene matching task. But after the videos were shown, you see this big increase in the probability of a correct match in the overlap, and to some extent a smaller increase in the no overlap and unseen conditions. Now, we asked people as well more extensively after scanning, to match the images, and it really was the case that in the no overlap and unseen conditions, they couldn't do it, right? So this increase that we're seeing here, we don't believe it's because people are figuring out which images were from the same location in these no overlap and unseen conditions. But if you think about it, one of these alternatives was in a no overlap, was in an overlap condition. It was an A1 or an A2 endpoint, and for that they could kind of effectively, if they knew that those images were from a different panorama, or they didn't belong to this scene or this location where the Q was, they could rule them out. So we think that this is kind of a recall to reject strategy is going on here, where you do see these increases in these two conditions. We don't think it's because of any kind of spatial learning. I think it's more likely to be that people are just recalling to reject some of the alternatives. Okay, and kind of the very first basic sanity check analysis that we did with the MRI data was a representational similarity analysis to look for patterns of activity that were specific to a particular scene, a particular viewpoint, and remained constant between sessions, so both before they had seen the videos and after. So we correlated patterns of activity before the videos had been shown with patterns of activity after the videos had shown. And the idea is that the correlation between like endpoints is higher than correlation between dissimilar endpoints. So, between endpoints A1 and B1 would be less similar than A1 sort of thing. And obviously, as you kind of expect, you see these big visual similarity effects in the primary visual cortex, spanning all the way down into the visual ventral stream, the right parahippocampal cortex, and the right retrosplenial cortex, which is good that we know we're sensitive to effects in these regions. I think that the left parahippocampal cortex came out as well, and but not the left retrosplenial. Okay, but for our main analyses, we really focused on 4 regions of interest, specifically the right parahippocampal cortex, the left parahippocampal cortex, the right retrosplenial, and the left retrosplenial, 4 a priori ROIs that were identified with this functional localizer. We didn't choose, although some previous studies had looked at the OPA, we deliberately didn't choose to look at that, which caused a bit of controversy at review. The reason that we didn't do this is because simply the BBB model doesn't make any explicit predictions about memory representations in that region. But also, I kind of knew we had some effect sizes from previous studies. We ran power calculations before the study started, and we knew that we were powered enough to detect effects in these 4 ROIs with applying a Bonferroni correction, but it just would have been too much to include any other ROI. So I specifically ignored the OPA. It turned out that the reviewers wanted to see it anyway, so we did an OPA analysis, and I can talk a bit more about that if you wish. during the questions. So yeah, 4 regions of interest: left and right, parahippocampal cortex, and retrosplenial cortex. Okay, so our basic analysis approach was to a general analysis approach was to think of every single way in which you might expect to see an increase in representational similarity between endpoints. And let me walk you through these different hypotheses for similarity increases, if you like. So firstly, you might expect that you'll see similarity increases between endpoints because they're from the same location. So for instance, across all conditions, you'll see a similarity increase between A1 and A2, B1 and B2, and C1 and C2. They're these just off diagonal cells of this kind of hypothesized correlation matrix. And, you might expect to see this because people might figure out which images were from the same location just by, experience with the endpoints themselves. Also, there are visual features that are shared across scenes from the same location incidentally. We know that participants were at chance in matching endpoints before the videos were shown. So that's good, but we still can't rule out that there were some visual similarities that participants weren't picking up on or weren't expressed in their behavioral performance. And we also did stuff like we used a model of vision, like the GIST descriptor, to kind of make sure that there was no obvious differences or similarities between images from different conditions and things like that. Simple luminance correlations and color content analyses, nothing showed up. But even then, we still have to be careful that we're not picking up on similarity increases that are just as a result that they are from the same location. So that's one kind of covariate, if you like, in the analysis. Another reason why you might expect similarity increases between videos are because the endpoints were shown in the same video. So kind of an associative memory effect, images that are shown in the same video might have some mnemonic association, and they would just become more similar as a result of that mnemonic. The representations will become more similar as a result of that association. And then finally, we have our main prediction. I called it here integrated views, but really it's just the overlap condition that image endpoints A1 and A2 specifically increase in similarity because they were shown in an overlap video. And it's not sufficient to look at each of these hypothesized effects separately, because you can kind of imagine if you have a sub-threshold effect for the same location correlation matrix and you have a sub-threshold effect for the scene together and add those together, they might look like an integrated views effect. They might look like the effect that you care about. Whereas including all of these terms in the model simultaneously in a model that explains the similarity of our data in the model, similarity of the data, sorry, you can be sure that you're attributing variance where it needs to be attributed to. So you're kind of explaining the data in the best way possible.
Sam, can I ask a question?
Yeah, sure.
I don't quite get the scene together bit. So C2 and B1
yes.
seen together. Does that mean that they're from the different panoramas, but it skipped from one to the other? Right. Okay.
Yeah. So they were from different panoramas, but they were shown in the same video. So B1 and C2 might be better over here. So this would be an example in a no overlap video. This would be an example of a B1 image because it's the 0 degree endpoint from this no overlap video, and the 180 degree image would be the C2 image.
And their task when they're looking at this is to decide whether they're from the same location or not.
So when they're watching the videos, it's just to detect a visual overlap between the two video segments. Right. That's what we asked them to do when they saw the videos. Right. When they saw the similarity analyses, I'm sorry if I missed this. The similarity analyses we ran on the scenes themselves. So this is when they were doing the red dots task, and they were just detecting those oddball targets. So there was no explicit memory or spatial task during that time. They were just we made sure that they were surveying the images.
Cool. All right, thanks.
No worries. Please, anyone feel free to jump in and ask questions at any time. I'm happy to field questions as we go through. So yeah, so we have these hypothesized increases in similarity represented by these similarity matrices over here, and we have the similarity in our data, which is basically a correlation matrix between bold patterns, Fisher transformed, and the idea is that we want to explain this the similarity in the data as a linear combination of these hypothesized similarity, and it's only if we can get an effect over here that this is our predictive effect. Basically, this is specifically what we're looking for. Anything else to say here? Yeah, just one thing to note: if you do this, you we had to be a little bit careful because the rows and columns of your similarity matrix the similarity in the data are by definition correlated, right? They're built from the same data, the cells in the rows and the columns. So we included a whole load of random slopes in a mixed effects model to kind of account for that similarity. The danger for that statistical dependence, the danger of not accounting for the statistical dependence between the rows and columns is that you might underestimate the error variance, and you will have too much confidence in the effects that you're detecting. So that was quite important to do. And obviously, like this, it was a mixed effects model across participants, so we had random slopes and effects for individual participants.
Last question. Is the integrated views, is that performance dependent then?
Very good question. I was going to come on to that. Yes. So no, we didn't have. It was a regressor in itself that was not dependent on performance. However, we also included a regressor that encoded your performance, your behavioral performance on the post-scanner task, and we calculated the interaction between the two, right? So we had one hypothesis, which was that we would see the main hypothesis was that we would see an increase regardless of your behavioral performance, which is what I'm representing here. In a supplementary analysis, if you like, what we did was we weighted this cell, the value in the cell, by how well they were able to recall identify which scenes were from the same location.
So how do you get why aren't all same location ones integrated views?
Because image B1 and C2, image B1 and B2 weren't shown in an overlap video. They were, and C1 and C2 likewise, they weren't shown in an overlap video. Yeah, I should say, even one thing to validate is that a good reason to do this is that we did find, even though we tried to match stimuli in terms of their gist descriptor and luminance correlations and color content and all of that stuff, we still find that the parahippocampal cortex bilaterally encodes visual information specific to the same location. So you do see a same location effect before they've even seen the videos. Yeah, so even though participants couldn't identify which images were from the same location before they had seen the videos, there was still some information in the parahippocampal cortex that meant that images from the same location were more similar to one another. So I do think it's quite important that we took into account this regressor. Okay, so what did the data show? So we only really saw things in the right parahippocampal cortex and the right retrosplenial cortex. We didn't see anything in the left hemisphere. I'm not going to make a big deal of that. We didn't do things like match. We didn't control the luminance of our stimuli or anything like that. So I'm not going to make any claims about laterality, but we did see effects in the right hemisphere and not the left. So in the right parahippocampal cortex, we saw the effect that we're interested in. And to be clear, this increase in similarity in the overlap condition after having seen the video, so post-video, you see an increase in overlap endpoints. And this was not dependent on their behavior. So it was just this matrix over here, this hypothesized matrix, not taking into account their behavioral performance, that we saw this increase over here. We didn't see any similar increases for the other conditions. The right retrosplenial cortex, we didn't see any kind of increase. This a priori hypothesized increase in in similarity, we did not see that in the retrosplenial cortex. However, when we did look at that interaction with the behavioral performance, so basically the correlation in the change of the similarity, this is overlap in the overlap condition. If you look at the change in the similarity pre to post videos, the change in similarity was correlated with how many matches the participants could make between images A1 and A2. So over here, let me just move my Zoom stuff. So over here, when they could match two correctly, that means that given image endpoint A1, they could correctly cue A2, and given endpoint A2 as a cue, they could retrieve A1 as a target in both directions. If they only got one correct match, that means that they could only do it in one direction, and if they got 0 correct matches, they didn't do it at all. And the degree to which they could do that task correlated with the number of correct matches, although it was actually only significant in this 2 condition. We just tested it as a linear effect. We look then, this raises the question, what happens? We found an effect in the parahippocampal cortex. Is that effect dependent on behavior? No, it's not. Right. So if you look for the same effect in the parahippocampal cortex, even in the zero number of correct responses, when that was 0 after scanning, so they were unable to match endpoints A1 and A2 together after scanning, even if they were completely unable to do that, we still saw the predicted increases in representational similarity. Now, obviously, that might just be because they had the association inside the scanner and they forgot it, or it wasn't very strong, so it wasn't able to come out in the data, or it was just noise. These slopes are statistically different from one another, and we weren't expecting to find regions that had any kind of statistical, any difference in slopes. We are kind of expected to either see flat lines or positive lines, but not regions showing a mixture of the two. We do speculate that there is a reason for this, and I should say now this is entirely post hoc. This is not a priori prediction that we made. However, we do think that the BBB model is able to account for this. And so I would like to think it's interesting. So I will talk about it very briefly. If you think back to our behavioral task, participants saw one endpoint as a cue, an egocentric view. And they had to, from that endpoint, they had to cue an allocentric representation, presumably. And from that allocentric representation, they had to cue another egocentric representation that matched one of these alternatives. So the task requires this forward and backward flow of information from the parietal to the medial temporal, right? And so if you kind of suppose that your performance is very heavily dependent on the behavioral task, is very heavily dependent on both the forward and reverse flows of information, you might expect information in the retrosplenial cortex to be more tightly coupled to your ability to do that forward and backward transformation. And maybe it would be less so. The information, the allocentric representation in the medial temporal lobe is less dependent on that. Because, for instance, I might be able to know that, pick out that this image of this church is opposite this building over here. But given this building over here is a cue, I might not be able to do the reverse. I might not be able to cue the allocentric representation given this image, even though I can do it with this one. So, that's speculation. But we think that we think it's plausible. I'd like to hear your thoughts on it as well, if anyone has any comments on that. Okay, so that's basically it. That's it for the main things that we wanted to look at. We did some exploratory tests to
Sam,
kind of yes.
Sorry to interrupt. Can I just ask a question about the model? If you just go back one slide.
Yeah, sure. Like, do you mean over here?
No, the slide that you just had on with the model.
Oh yeah, sure.
Yeah. You talk a lot about the parietal, the circle with the parietal representation, the egocentric representations of the scenes.
Yes.
But you're not looking at any parietal regions. And
yeah.
Wouldn't the classic view be that the PPA, which is this essentially visual representation, I assume that falls in the sensory cortex part of this model.
Well, the parahippocampal cortex, it supposedly encodes geometric information. In the model, they say, for example, it encodes positions relative to spatial boundaries. So it's considered part of the allocentric reference frame, the parietal representation. The parahippocampal representation. Is that what you were saying? The parahippocampal? Yeah,
I guess I've my last interaction with this literature. I thought that the PPA was considered to be an egocentric representation, but anyway, that's probably well out of date anyway. But I guess my main question is, why are you not looking at any parietal regions?
Good question. I think so. So the reason is that we would have to do a different experiment, or we would have to at least use different stimuli. So to look at, to separate, to identify egocentric representations, you might need to show the same egocentric view, but change some visual properties. Like you might need to show only the high spatial frequencies in the image, because that is still presenting some egocentric information, but it's kind of different to the low level visual information that you might be seeing, or you might, show it on different days or under different lighting conditions. In general, I think it's much harder to make a task where you're looking at, you're trying to identify specifically egocentric representations in these parietal cortices, because they're also correlated with a lot of low level visual features of the image. So it's quite difficult to separate those two things out. And we only used one egocentric view for in this experiment. So we couldn't really do an analysis where we say, oh, this is where we think the egocentric representations are, because we couldn't separate that out from just purely edge detectors and stuff that you would see in primary sensory cortex. And obviously we did see, so if I go back to over here, this is the scene specific patterns specific to a particular egocentric view, and obviously we do see the parietal cortex. Interestingly, in the model, especially in the 2007 BBB model, they don't really talk about which regions are involved. It's just the parietal cortex, but some people have ideas of where it is exactly, and we do see some parietal regions. You can go onto the link here to see which regions are there, but we don't know whether they're just purely visual edge detectors and stuff like that, or they are actually representing the egocentric information abstracted from those low-level visual features.
Cool, thank you.
No worries. So yeah, as I say, exploratory analyses now. These were we recorded brain activity as participants were watching the videos, but we didn't have any hypotheses. So this is truly all exploratory, and we just did a simple bold contrast, overlap versus no overlap. So we found higher levels of activity while participants were watching the no overlap video in the parahippocampal cortex, and all the way actually in sensory regions as well, kind of late sensory visual cortices as well. And you might think, oh well, why is it higher for the no overlap videos? And I'm ashamed to say I didn't see this at first. I actually gave this talk at an EPS meeting, and I said, oh well, maybe I think I had just had the data and I just got it off the press, and I was just going, well, why could it be this? Could it be like so we don't know how the bold effect maps onto memory encoding. Maybe it's like a memory encoding effect. And then Neil Burgess was in the audience, and he said, well, it's just adaptation, right? Because the the overlap videos show that central 90-degree region of the panorama twice. The no overlap videos do not do that. They're different things. So there's a repetition in the overlap videos, which would lead you to believe that there's some adaptation, or it should cause some visual adaptation, and that's what we believe this no overlap effect is showing. A no overlap greater than overlap. Again, it could be related. So it could just be a release from inhibition. But we do see this overlap greater than no overlap bold effect in the medial prefrontal cortex in one big cluster. I think it's one big cluster, and it's a little hard to explain, but yeah, it could just simply be a release from adaptation. It could be related to the behavioral task. Participants were asked to identify whether there was an overlap in the panoramas. So this could just be something to do with the demands of the task. But alternatively, it could be something to do with some sort of memory integration. Again, I have no evidence other than what you see to say that. So yeah, I would not make any big claims about that at all. But you do see the overlap or the no overlap condition difference became obvious 5 seconds into the video, and then kind of if you build an HRF, you can kind of see the separation between these two lines at about the right time. Okay, so just to sum up, we showed that there was these rapid learning of location-based allocentric representations in the parahippocampal cortex and the retrosplenial cortex. We think that this is consistent with the BBB model. And also consistent with the BB model, it's these representations were activated during passive viewing, where there was no explicit spatial task in force. We find this non-hypothesized dissociation between the parahippocampal cortex and the retrosplenial cortex, in terms of, the retrosplenial cortex is more predictive of behavior than the parahippocampal cortex. We think that could be accounted for by the BBB model, but it was not a priori prediction. And we've seen that the medial frontal regions are engaged during spatial learning, although they might not be anything to do with it at all. So yeah, I think that's it. Thank you to my former supervisor at York, Aidan Horner, and the PhD student I mentioned who collected the stimuli, Bardor, who is now a postdoc with Neil Burgess at UCL. And thank you all for listening.
Thank you very much, Sam. That was fascinating stuff.
Thank you.
Does anybody have any questions?
I've got a question. It's a slightly nasty question.
Go ahead.
But, well, maybe it's not. So I don't have very good allocentric spatial memory, I don't think, and certainly in tasks like this. And when you were showing the videos, I kind of thought, if I was doing this, I'd probably be trying desperately to name the things that I'm looking at in these videos, and then seeing whether, I could recognize the endpoint, whether it matched up or not. So how do you know that people aren't just trying to look at sequences of items, and those items happen to be scenes, and so you're engaging scene processing areas, but you're effectively just kind of racking up 3 items that you see going one direction, and then 3 items you see going the backwards direction, and if you end up with the same item, then you think, oh, I better remember this goes together.
Yeah, absolutely. There is no guarantee that the videos that we produced for the study were sufficient to form an allocentric representation, or I'm going to be careful, even just a viewpoint-independent representation. There's no guarantee that happened, and so this project was very risky in that respect, in that a null result means absolutely nothing, because there's not much that you can say with a null result. But you would expect that an allocentric representation would lead to this similarity increase in these regions that we predicted a priori, if the videos were sufficient to elicit one. So I think it's one of those things that the model made a prediction for these effects, but there was no guarantee that our stimuli were sufficient to generate that representation that we so really it really was the case that a null result probably wasn't interpretable.
I guess that there's different ways of testing a model, right? And one way would be to you've kind of tried to get evidence to support that model, but you could gather evidence that would end up showing the same effect, but then would not be consistent with the model. So just because you found that supportive evidence, like, I'm not sure it's great support for that particular model.
No.
Maybe a simpler explanation that would also lead to the same results.
I agree that there are other explanations. So in the paper, there are a couple of alternative explanations that we talk about. So one of them, the one that I like quite a lot, is that it could be attentional, right? So like when you see image A1 for the first time, you don't know which features are shared between A1 and A2, right? But after you've seen a video, maybe you start to recognize, oh, well, it's the lampposts that are the same in A1 and A2. So for instance, Brad Love has a model where, this kind of attentional learning is really important for recognizing objects and discriminating objects and things in our environment. And so, it might be nothing to do with that. So I completely agree that it's not necessarily, we can't beyond doubt say that these representations, these viewpoint-independent representations, we'll call them viewpoint-independent representations. We can't beyond doubt say that they're allocentric. I entirely agree. That is a doubt. But we did, I guess, we lend support to the model because the model was the thing that inspired the predictions. But yeah, I agree that you might disagree and say, I actually think that these other models are better able to account for it. And I think that the Brad, this attentional thing is probably is very able to account for these data, and maybe it is more simple. I'm not sure, but
I mean, it's a very neat effect, and so I should have said that first. But it's very nice to.
No, it's all good. So I think that's an area for future research. Like, is it really? Are these effects that we're identifying really allocentric in nature? I think that is the core outstanding question that we have. We believe that this is support for a model. We do believe it supports the model, but we don't actually have any direct evidence that these representations that we're picking up are truly allocentric in nature. And there are some things that we could do, like DCM, to try and look at the connectivity between different regions to say, well, is the connectivity between regions supportive of the model, and try and test the model in different ways.
Does anybody else have a question? I have one more question.
Sure.
Do you have your you've done your RSA and you've tested that within these regions of interest. Is it possible with your analysis to do a kind of like Searchlight RSA and look for the three components of the model and which brain regions they map onto in like a, I don't know, like a winner take all kind of thing, and then make a whole brain map of which parts of the model best is that? I think that's possible, but and have you done that, I guess is the question.
Well, no, I haven't done that. As I kind of said like before, we can't really discriminate between discriminate between visual representations of specific scenes that are basic line and edge detectors and things that we think contribute to this egocentric reference frame, where it's really kind of encoding the position of objects relative to the agent in the egocentric frame, or the position of boundaries relative to the agent. So we can't kind of, with these data, we can't separate those out. We could, well, yeah, even then, like with the analyses that we've done, we can't even discriminate between egocentric information that is activated that is activated as a result of this. So for instance, let me explain what I mean. Image number 3 is actually the corresponding image to this target, right? It could very well be that when you're in the scanner and you're seeing this church, you're activating an egocentric representation for the other field of view, right? Not an allocentric representation, just the egocentric representation for number 3, even though you can't currently see it. And so actually we're picking up on egocentric representations. This is why we try to refer to them as not really allocentric representations, but we're detecting viewpoint-independent representations that we believe lend support to the model. We don't really know if they're allocentric in nature. So I don't think that the paradigm gives us enough kind of leverage to separate different components of the model all that well. We just used it to generate these predictions.
But you can separate the 3 components within a given region.
It's not obvious to me.
Yeah, your results kind of give you a different answer for each of the 3 components of the model, right?
Yeah, I mean, so the analysis that we did, we can't say, like, it's not sensitive to egocentric representations of the same view of a view. The analysis that we ran, our main a priori hypothesis analyses, they're not sensitive to the to egocentric representations. They're only sensitive to these viewpoint-independent representations. So we're only really sensitive to these, what we think is going on here, and maybe in the retrosplenial cortex, what's going on here.
So I guess like, can you not build a searchlight that detects viewpoint invariant representation?
Oh yeah. We could do it as a searchlight, yes.
Yeah, and then I guess if you did that and it showed, like, you would hope it shows the same regions that you've chosen as regions of interest, but might also show other regions, and that might also kind of like lend additional support to this model, or or allow you to do some more differentiation between different models.
Yes,
it's a thing that you could do and you haven't looked at, that maybe would allow some more theorizing.
Yes, absolutely. Our searchlights weren't, like, we didn't test them, we take them seriously because we didn't think that we would have the power. However, we did do a searchlight analysis for the RSA effects. It's in the paper and on the OSF. So yeah, you can go download them. They show similar results in the regions that we talk about, and but nothing really significant at the whole brain level.
Cool. Great stuff. I think we've come to the end of the hour, so thank you very much, Sam, and thank you very much everybody else for coming along. Next week we have some project proposals, so I hope to see you all then.
Great, thank you very much again.
Yeah,
See ya.